So one announcement before we get started. So the faculty software interface failed over the weekend, and so I haven't gotten you your information about your teams and your information for how to access the questions for your review of the material from the weeks before. Hope I'll have that done by the end of the day today or by tomorrow at the latest. And if the conventional software interface I can't master right away, we'll figure out a way to work around that. So I'm sorry for the delay, and I'm sorry I didn't communicate about that. It was the out of town over the weekend syndrome. So, so it's terrific to have today Dr. George Howard as our speaker. He's a professor in the Department of Biostatistics here at UAB. Just a real bit of brief history. So he did most of his training and early career at the University of North Carolina. Came to UAB, I think, in 1999 as department chair for biostatistics and served in that role till around 2011. And as you said, he has the best job in the world now, ex-department chair, which I can relate to. His research has been wide-ranging, but really probably the highest impact and still very, very active is that he was the originating PI for the regards study. I can never remember exactly what regards stands for.
Reasons for geographic and racial differences in stroke.
Right.
You can't do research if you can't do acronyms.
Right. And this is a huge national study that has, I don't know how many times it's been renewed, must be 3 or 4 cycles now. It has an incredible amount of data that's been used well beyond the stroke aspect of the original intent of the study, and Dr. Howard stays as the a major user of the data, but also as a national coordinator for access to that information. You know, I think if you have any interest in accessing information from large countrywide research databases, he's a terrific person to talk to about how to make that happen most smoothly and what issues you have to confront, how you deal with the IRB issues, how you deal with all of the stuff associated with accessing data on a big trial. He's also the director of a national database on, it's called CRESS, which is, has to do with revascularization of carotid arteries for stroke prevention. And is just a great resource in biostatistics. And he's going to talk today about phase 3 clinical trials and beyond.
Thank you.
If he stands here, we don't have our lapel mic today, so if he stands here, he'll be easily heard by the people online, and I'll have this microphone to walk around with. The people who are joining us online can hear faintly if you talk in a normal voice without the microphone, but if you really speak strongly with a normal voice, the microphones in the ceiling work pretty well. So if you have a comment, just please speak up. I may need to give this to Dr. Howard because he likes to roam, and we should get to do that. Okay.
No, I'm honored to be here. Please, I can't stand lectures, and please consider this a discussion more than a lecture. Please feel invited to stop me at any time. There are a ton of slides, and if we don't make it to the end of the slide deck, I consider that good, not bad, because that means we've discussed and gone into more depth on other things. And so please absolutely feel invited to stop me at any time. I'll also say that this lecture is a mess. Basically, this is a semester-long course in the School of Public Health taught in one day here. And so you will have the feeling that it is wandering through 1,000 different topics and jumping from topic to topic. And the reason for that is we're going to wander through 1,000 different topics and jump from topic to topic. And so basically, if you seem confused or something, just slow me down. Slow me down and we'll figure this out together. Basically, there's a lot to cover in something as simple as clinical trials. They really are simple. That's one of the beautiful things about them. And so basically, I'd like to put this in a little bit of context. And so basically, if you consider what what is the spectrum of evidence? Why do we believe what we believe? And basically, studies can be sort of ranked as from the weakest to the strongest. And clearly, the weakest are what are called ecologic studies. And ecologic studies are that people in the south of France drink a lot of wine, and people in the south of France tend not to die of heart disease. Diabetics because they drink a lot of wine in southern France. And the key of an ecologic study is I didn't measure anything on anybody. I measured outcomes for a group of people and exposures for a group of people, and then I guessed that they are associated. Sort of moving up the food chain, the next level is observational epidemiology, something dear to my heart. And within that, there's also sort of a ranking, a food chain of studies. And I wish I had more time to talk about all of these, and I do hope they're covered in your experience. But sort of the weakest is a case control, where I get sick people and well people and look backwards to see what made them sick and well. The next level up is a cross-sectional design. You can think of that being a snapshot. I take a picture at one point in time and see who's sick and who's well and who's exposed and who's not exposed. There's no temporality in a case control study. The highest on the food chain of observational studies is the prospective cohort. And basically the idea here is I get a group of people who generally don't have the disease, and I measure a whole bunch of stuff, and then I wait patiently, sort of vulture-like, to see who develops disease and who doesn't get the disease, and then say, aha, it was the people who smoked who got lung cancer later. And so that's it. And then finally, is this what we're talking about today, the randomized clinical trial? It's always referred to as the gold standard evidence. And it's amazing how few people don't really understand why it's the gold standard evidence. Wanted to take a second and make sure you're cool with that. And so basically, in the 1990s, I'm old. In the 1990s, there were a boatload of beautifully done studies that clearly demonstrated hormone replacement was protective of heart disease and stroke in women. In fact, I stood at the podium of the Heart Epi meeting and put my arm around Trudy Bush, God rest her soul, and said, we can never mount the Women's Health Initiative because we all know for sure that hormone replacement protects women from heart disease. It is unethical to do this study. And then 3 clinical trials with many fewer patients in the observational studies suggested hormone replacement was not only protective, in fact, it was harmful. You know, the entire field just dumped those epi studies and went with the clinical trials. It's now dogma that hormone replacement's not cool. How can that occur? Why do we accept the results of the RCTs over the results of those incredibly well done, beautifully done observational epi studies? And so the idea of this is an important topic for today, which is confounding. I want to make sure, by the way, at different points in my lectures, I always say, this is an important point. I'd like you to remember that. I'll say a bunch of stuff that must not be important because I don't say it then, but this is one of the things I'd like you to take home. This is one of the things so basically a confounder is I have some exposure, estrogen, and I'm interested in seeing if it's related to some outcome, heart disease. Okay, so that's the relationship I'm interested in. Okay, but I've got this other thing hanging around in the ether called a confounder, and it's for this is probably socioeconomic status. Probably socioeconomic status. And it turns out that rich white women took hormone replacement. I mean, rich, and also rich white women don't have heart disease. Okay, and so instead, what we were fooled into thinking was that by the way, people define things differently in science. I'm sure you've noticed that. My definition of a confounder is anything that's associated with both the risk factor and the outcome. Okay, if you're a hardcore epidemiologist, there's a little phrase that is added that says, and it's not in the pathway. Okay, I'm not smart enough to know what's in the pathway and what's not in the pathway. And my example of that is always inflammation. Inflammation is causative probably of atherosclerosis. And by the way, atherosclerosis is an inflammatory process that increases inflammation, so the things are pointing at each other. Is it in the pathway or not? I don't have a clue. My definition is anything that's associated with both the risk factor and the outcome, and these can lead to false associations, and that's what happened here. Okay, and so in this case, rich white women are more likely to take hormone replacement, and rich white women are less likely to have heart disease, so it looks like hormone replacement protected you from heart disease. Okay, so how does randomization fix confounding? Well, with randomization, the high SES women and the low SES women were equally exposed to the risk factor. Okay, and so basically that leg of the three-legged table is broken. Okay, and it doesn't matter that the confounder is still related to the outcome, because things cannot be a confounder unless they're related to both the exposure and the outcome, and randomization breaks that three-legged table, and that's cool. That's cool, and remember this, that's the cool thing about randomization. Now, randomization is wonderful for many reasons. I wish I could talk more about it. But the single biggest reason is not only are high and low SES women equally exposed, but there's equal exposure for all risk factors. For all risk factors, even the ones we don't know about. It fixes the confounders for factors that you're not even aware of yet. It turns out observational epi, and I think I say this here. So I don't know if I say this, I might say this in a minute. I've forgotten what I'm doing next. But anyway, so the cool thing about randomization is that it does it for both the ones you know about and the ones you don't know about. And so why not do all randomized trial? And the reason for that is research is a process. You start out with ecologic studies, you move through randomization, and then finally at the end of the day, when it's really well understood, you do the trial to put the nails in the coffin. You don't start by doing a randomized clinical trial. They're really expensive. The CREST trial that was mentioned in the introduction is a $65 million study, and you don't frivolously do $69 million studies. And basically, they only address one question at a time. So if you think about it, that's a boatload of money to answer one question. And also, some things just can't be done with randomized clinical trial. We all believe seatbelts prevent deaths in cars. There's never been a randomized trial of seatbelts. Interestingly, the tobacco companies, I used to be from Winston-Salem, a town, the cigarettes were named for the town, not the town for the cigarettes, by the way. But anyway, and the tobacco companies are so cool. They come out and they say, there has never been a definitive evidence that cigarettes cause lung cancer. And I agree 100% with them. Absolutely a true statement, okay? To do that, you would need to randomize children to smoke and not smoke, and then follow them forward and wait for who develops lung cancer. And then we would have the proof that cigarettes cause lung cancer. And the people who buy into this, I don't believe it if it's not a clinical trial, should be comfortable with the tobacco company's claim, correct claim, that there's no definitive evidence of a link, because they're right, okay? But anyway, so a lot of things can't be done. Parachutes, never been a randomized trial of parachutes. It's just not going to get done. Okay, so in terms of the big picture of trials, and all this is just leading in, I'm talking about phase 3 trials, and it's important to put those in the context of the clinical trials process. And so the first trial is called a phase 1 trial. By the way, this phase 1, 2, 3, 4, different definitions by different people, so watch out. These are my definitions, and most people agree with me. Other people have different definitions. A phase 1 trial is simply, is the drug tolerable? The first in humans attempts. If I give this to somebody, do they die on the spot? Typically done, particularly in cancer trials, in very sick people, just to make sure the drug is cool. Okay, the next is where we begin to get different definitions. A phase 2 trial, early efficacy trials. And they're usually divided into what's called phase 2A and 2B. A phase 2A trial is a safety dosing study, usually giving the drug and looking at the levels of the drug in the serum. You know, are we delivering, because if you think about it, where we're doing is moving from giving drugs to rats to giving drugs to people. And if you start trying to guess, well, you know, rat's about that big and I use this much drug and the human's about this big, we're just going to multiply the height of the rat by the height of the person, which is pretty much what you do to get the dosing for the person, okay? Might guess wrong. So the first level is safety dosing. The second is early efficacy assessments. That's phase 2B, okay? Early efficacy assessments look at surrogate outcomes. Okay, so I'm not going to look to see if we prevent stroke. I'm going to look if we prevent stroke on a CT scan, okay, which is a subclinical stroke. So I'm going to look to see if it stops progression of carotid atherosclerosis. Because if a phase 2 trial had the same outcome as a phase 3 trial, it would be called a phase 3 trial, okay? Then finally, what we're talking about today is the pivotal trial. Does safety, efficacy, meets regulatory requirements? Importantly, it doesn't answer all the questions all the time. By the way, y'all should be asking questions too, so stop me. Remember, I wanted you to stop me. Anyway, so next is the post-marketing assessments, the, you know, further safety trials. And so why don't we nail down safety in the phase 3? Well, the phase 3 trial is powered to go after efficacy. Does the thing work? It's not powered to go after safety. Is this thing safe to use? So the example of that, perfect example, is Vioxx. Okay, Vioxx, incredibly efficacious, stops pain, works great, okay, but it increases heart disease risk. But the heart disease risk is still so rare that the study's not big enough to see it in the phase 3 trial. And so we have to do this post-marketing assessments of safety afterwards. And so this is really all a process, but for today, we're just focusing on the phase 3 trial. Everybody cool? Yes, ma'am. Thank you.
In phase 4 trials, when you say post-marketing, is that like the FDA gives like, okay, we start seeing commercials for it and there's still trials going
Yeah,
on?
exactly. The question was, is this something basically required by the FDA? And so exactly, that's precisely what happens. The FDA approves the drug, but says, oh, we're a little bit worried about heart attacks with your Vioxx. And we're going to require you to collect data systematically to make sure that people aren't dropping dead while their knees feel great. Okay? And so basically, usually they're required by the FDA. FDA is incredibly, phenomenally powerful. Someday you will have an FDA audit and you will be impressed by how powerful your FDA is. It's an amazing process. Okay, so here is the outline for today. This is all we need to cover today, okay? And this is why it's going to seem like we're hopping around a whole lot, okay? And when I teach this as a course, each one of those brown things is a separate lecture. And so what we're going to do is 3 slides summarizing a whole bunch of a semester-long lectures, okay? So don't get too confused. Try to bear with me. We're going to start off with the ethics of clinical trial. It's really hard. I mean, if you think about it, all a clinical trial is human experimentation. That's all it is. Okay, we're randomizing people to different treatments. The barrier between us and the Nazis is phenomenally small. I mean, it is phenomenal. I mean, stop and think. If you're a clinician, most of you, I assume, are clinicians. When you're asking somebody to be in a trial, that is a huge ask. I mean, that's not a small thing. And the key to it is equipoise. And a lot of people don't understand equipoise. I mean, are any of y'all surgeons in the group? Oh, I get to insult surgeons. They're online. Okay, I'm sorry. Please forgive me. I've never met a surgeon who's at equipoise. I mean, these guys and gals know with certainty that cutting you open is going to make you better. I mean, they don't think that. And by the way, I want them to think that. I mean, that's not a bad thing. That's a good thing. If somebody is cutting me open, I want him or her to have a very deep belief that they've got a good reason to do it. Okay? Equipoise is not whether I doubt it, it's whether we doubt it. Whether, in your opinion, there's not a collective body of evidence that we share and agree on that this treatment is good. Equipoise is a community statement, not an individual statement. And you can remember that, again, because surgeons are never at equipoise. They're never at equipoise. Okay, again, clinical community versus, oh, by the way, I love how we do trials and how the Europe does trials. They have a different principle. They deal with what's called the uncertainty principle. And the uncertainty principle, if you do a European trial or you do trials with Europeans, they put people in studies when the individual PI is uncertain what study is best, what treatment is best. Okay, fundamentally different approach. You know, in the U.S., we get all over sites who don't put eligible people in studies because we worry about whether it's generalizable to the entire group. And so if we find out that you're not putting somebody in the study who's eligible, we come down on you like a ton of bricks. That's different in Europe. They say, oh, I wasn't sure. I was pretty sure, this treatment would help the person. I'm not going to put them in. So that's not a subtle difference. Okay, and that brings up the whole topic of informed consent. And how many of y'all have consented patients for studies? So everybody has. Do you think they understood what you were saying? Tell the truth. I mean, here's the cynical comment for today. Informed consent is to protect the university. It is not to protect the patient. I mean, regards, which I was brought up at the beginning of this, the most invasive thing we do is draw blood. That's the worst thing we do to people. Our informed consent is 8 pages long. Can you believe? I mean, that's amazing. The worst thing we do is stick a needle in their arm. Anyway, and so stop and think. I think we do have a duty to have people understand because what we're asking them to do is, as I said, a huge ask. But in general, that's going to take more than going over the informed consent document. I think it really will require a discussion with these people that goes quite beyond that. So the next topic, if you look at that outline, was the different type of hypotheses. Okay, and so basically, what I want to do is have a hypothesis that's clear, quantifiable, simple, singular. I'm doing one thing in a clinical trial. And what it does is give the who, what, and when of the study. This is the CREST primary hypothesis, and it's to contrast the relative efficacy of stenting and endarterectomy, that's the treatment, and preventing the stroke, myocardial death during a 30-day procedural period, and stroke afterwards. That's the what and the when. And then in patients with symptomatic or asymptomatic extracranial stenosis, that's the who. So when you're thinking about writing a hypothesis, make sure you've got the who, what, and when are all there. And it's amazing when you read these from even big trials, a lot of times, you should be able to give your hypothesis statement to somebody and have them pretty much understand what you're doing. They won't understand the details, but they'll get it, the 30,000-foot view. Okay, now let's get into something that is a little bit more complex, which is, and people talk about superiority trials and inferiority or equivalence trials. And this is sort of an important thing and a different way of thinking. So I'd like to take a few minutes and just go over those and make sure that you're cool with this. And so basically the idea of a superiority trial is to address if 2 treatments are the same. So by the way, in all of statistical testing, whatever you want to prove is true, you assume is false. Okay, then you go out and collect data and try to prove how silly you were in saying that it was false. That's the whole idea of statistical testing. So if you're doing a superiority trial and I'm trying to prove 2 things are different, I assume that they're the same. I assume that whatever I'm trying to prove is not true. I conduct an experiment to prove how different they are. And, you know, basically what I have is, suppose there's a new treatment and an old treatment. And I want to show the new treatment is as good, really not worse than, the old treatment. That's a whole different way of thinking. That's a lot different than proving that they're the same. If I want to show that I'm setting out to prove they're as good. So let's say the new treatment is less invasive and costs less. Okay, so I don't need to prove that it's better. I only have to prove that it's as good. Because if I'll save money and I won't have to do surgery, for example. Yes, ma'am.
So this is always confused me with drugs. If there is a new drug and the randomized trial at the end compares that drug to the standard of care, and there is no difference, but the drug, for whatever reason, costs less, the FDA can then still approve it, even though there's no benefit to the patient.
This is a great question, and this is exactly what I'm talking about. This is what this, I tell you, let me get to the end of this. So the question was, if you do a trial and there's no difference, can the FDA still approve it and claim that it's as good? The answer to that is, it depends on what their hypothesis was. Okay, and that's what the difference that I'm trying to bring up here is. And so basically, and this is important, if you're doing a superiority trial, trying to prove two things are different, I assume that they're the same, and the trial, I hate this word, it's a negative trial. You don't see a difference. There are two reasons why it could be the same. The first reason is it's the same. Okay, it really is the same. And the second reason is I didn't have enough people in my study to see whatever difference was really there. Okay? There's no way on earth you can know which one of those is happening. Once you do a superiority trial and there's no difference, you can never know which one of those two things happen. It's impossible. Okay? So basically, you cannot tell which is true. One never, by the way, in all of statistical testing, this is one of those take-home points. One never accepts the null hypothesis. Okay, one only fails to reject it. Let me say that again because that one's important. In all of statistical testing, you never accept the null hypothesis. You would never say that they're the same. What you would say is that, by the way, this implies that traditional studies cannot be used to show that the new product is as good as the old. So if it comes out negative, what you say, and this is on the next slide, you end up with sort of a kiss your sister kind of statement. There's not sufficient evidence in these data to show that the two groups are different. Intentionally a double negative statement. Let me say that one again. There's not sufficient evidence in these data to show that the two groups are different. Okay, that's a whole lot different than saying they're the same.
Can I ask you a question about that last slide?
Sure.
So you said you can't tell the difference between if there's no difference or the study size is the sample size isn't large enough. What if you have a very large sample size?
Well, it's still not large enough. By the way, there are no two things in nature that are the same. Okay, the intelligence of men and women is not the same. A radical statement, get me in trouble. I don't know which one's smarter, but the chance that there two things are exactly precisely the same is impossible, statistically impossible. There is nobody 5 feet tall in the world, okay? We just haven't measured people well enough. If we keep measuring them with sufficient precision, we will find out this person is either a smidgen above or a smidgen below 5 feet. There's nobody 5 feet tall. Okay, the philosophical part of this is that means the null hypothesis is always false. The two groups are always different. Okay, I just can't tell in what direction or how big it is. And so that all that is to answer, if the sample size was big enough, I would have found the difference that I know exists with certainty. But that's sort of philosophical, statistical philosophy instead of anything else. Okay, so basically, you know, so the answer is, if I want to prove two things are the same or one is as good, what I have to do is to change the form of my hypothesis. I have to use a different form of my hypothesis. Okay, and so instead, and this was work by Blackwelder, frequently also called Blackwelder trials of superiority or non-inferiority trials. And so the new hypothesis is the new treatment is worse than the old. That is my null hypothesis. And my alternative hypothesis is now the new treatment is as good or better than my old. I've turned the hypothesis inside out. I want to leave this up here for a minute because I do see some wrinkled brows. And does everybody understand that what I'm doing is playing a game with the hypothesis? I'm changing to what I want to prove is true, I assume is false. If I want to prove it's as good, well, I'm going to assume that's not true. That means that the new treatment is worse than the old. And now I'm set up to prove it is as good or better than the old. Pretty cool, huh? Cool idea. Really backwards thinking, okay? And so if we reject the null hypothesis, then we can make a definitive statement that the new treatment is as good or better, because I can accept my alternative hypothesis in statistical testing. I just can't accept my null hypothesis. Everybody cool? Okay, so simple in concept. Non-inferiority, sometimes referred to as equivalency, sometimes referred to as Blackwelder trials. There is a slight difference between those. Don't worry about it. At the level you're at, just think of them being the same. Okay? And so then basically, regardless of the form of the hypothesis, a study provides an estimate of the difference between the treatments. I'm going to do a trial and estimate how different the two treatments are. I'm going to calculate 95% confidence bounds around those estimates. Here's a cool thing. There's a link between hypothesis testing and estimation. And basically, if whatever I hypothesize the difference is inside of the confidence limits, I will always fail to reject my null hypothesis. Alternatively, if whatever I'm assuming is outside of the 95% confidence limits, I'll always reject. And I'll show you what I mean by this. Right now, I see more crinkled brows here, but you'll get this in a minute. So basically, so what I'm going to do is a little bit of background. Let's say I have two treatments or products. I have an established treatment and a new treatment. I have a dichotomous outcome, a yes-no outcome. And I just want to calculate the percent of events in the established treatment. I'm going to call that pi sub 0. I don't do a lot of math, trust me. Don't panic if you're a math phobe. Then the number of events in the alternative treatment, pi n, and in general, which treatment is best. So, traditional hypothesis testing, superiority testing, traditional, okay? The null hypothesis, no difference between the treatment groups. So I'm going to say that pi 0 is equal to pi n. Okay, alternative hypothesis is the groups are different. Okay, that pi 0 is not equal to pi n. This is not hard, by the way. Even I can do this. This is easy. Okay, so basically, if you do that, then what I have here is a bar, and on this side of it, I'm saying the old treatment is better. On that side, I'm saying the new treatment is better. So what I do is I calculate the difference in those two proportions. And I can put a little bar on my graph and say, there it is. Okay. And then I can calculate 95% confidence bounds around that. I can draw those. Okay. Now, if that happens, if the 95% confidence limits do not include 0, then the two groups are different. That's how simple traditional testing is. So basically, if you think about it, there are three things that can happen if you do a traditional superiority trial. Okay. You can claim that the new treatment is better than the old. You can claim that there's no difference between the two treatments. Or you can claim the old treatment is better. Okay, those are the two things. And by the way, I just fundamentally messed up. I said you can claim the two groups are the same. I should have said that was a slip and a bad one in this class. Okay, what should I have said? No evidence in these data that the two groups are different. And by the way, going back to your sample size question, basically if you increase the sample size, you keep making those confidence bounds itsy-bitsier. And if I keep on making them itsy-bitsier, I will always find differences. We're going to come to how itsy-bitsy you want them as part of this lecture, by the way. Okay, so what are the advantages and disadvantages of a traditional study? The advantage is I can make a definitive statement that one treatment is better or worse. Okay? The disadvantage is if there's not an evidence of a difference, I cannot say that the two treatments are the same. I should not do that. Okay? The null hypothesis is not, by the way, in a lecture, when anybody gives a lecture and they say the same thing like 20 times, that's because they think it's really important. Okay? So the null hypothesis is never accepted. It's simply a case where you fail to reject it and you end up making a statement there's not sufficient evidence in these data to establish that a difference exists. Okay? And but the manufacturer sometimes only wants to say that the new treatment is as good. And if you do that, you do an equivalency analysis. Same picture. The goal here is to make a definitive statement that the new treatment is as good or better than the old. The null hypothesis is now the new treatment is worse than the old treatment. And here's the twist, plus a fudge factor. Okay, plus a fudge factor. And so basically what you say is that pi sub 0 plus the fudge factor is less than the new treatment. Okay. And the alternative is that the new treatment is as good or better than the old treatment plus the cushion, okay, which is the pi 0 plus the fudge factor is bigger than delta n. Okay, and so basically, therefore, the definition is as good really means that it's within the cushion. Every superiority trial you will ever read has a fudge factor. When you're reading these, you should be, by the way, in the discussion, if it comes out as a positive inferiority study, they say the new treatment is as good or better than the old. What they really should say is the new treatment is as good or better than the old, and I define as good as being within 2%. Okay? Okay, that fudge factor is always there. If you're reading a superiority, non-inferiority trial, always be acutely aware of that fudge factor, how big that fudge factor is, and whether you think being within that fudge factor means it's the same. So, testing between groups in the equivalency approach, what I have is the new treatment, the no difference, here's my fudge factor, and I can calculate the difference in the proportions, there it is. And then I calculate, and here's the other difference in superiority testing, a one-sided confidence bound. And so what I'm going to do is calculate a one-sided 95% confidence bound around that estimate, and it starts there, it goes up to infinity in that direction. Okay? And if you do this, two things can happen. Oh, by the way, if the 95%, the bottom end of this 95% confidence bound right there, that dark blue thing, does not include delta, okay, then the new treatment is as good or better than the old. Because really, if you think about what we're saying, let's see if this works again. Okay, really what you're saying is that I'm pretty sure the treatment difference is in that blue line, and if this is what I declare as no difference, if it's above that, then it must be as good or better than the old. And that is the whole idea of superiority testing. Is everybody, I see some sort of confused faces, so is everybody okay? Any questions or anything? This isn't hard. Don't make this hard. Make this easy. Okay, so the outcomes in equivalency testing, only two things can happen. You're either going to say the new treatment is as good or better than the old treatment, or you'll say there's not evidence that the new treatment is as good or better than the old treatment. Because in this case, the bottom of this confidence bound includes the delta.
Is delta the fudge factor?
Delta is the fudge factor. In carotid endarterectomy trials, your FDA has pretty much defined the same as being within 2%. And so if I'll ask you, if I was your patient and you were going to do a study on me, and I had a 10% chance of dying on the table versus a 8% chance of dying on the table, would you consider that the same? Okay. Now, this delta is tightly tied to the sample size delta that you're talking about, which is defined as a clinically significant difference. We're going to come back and talk about this later. And basically, they're the exact same concept. Okay. In other words, if you're designing a superiority trial, you have to say, I'm designing this trial and I'm interested in detecting differences bigger than 2%. Okay.
How is the delta determined? Is it based on pilot data or?
Okay, so the question was, how was the delta determined? Was it based on pilot data? It's interesting. If you think about it, if you're doing a pilot study to estimate what the difference is, by definition, you don't have enough people in your study to well estimate what the difference is. If you did, you wouldn't be calling it a pilot study, you'd be calling it the study. Okay, and so the one place that you really shouldn't get the difference is from pilot data. Because you've just said, I have an unreliable study that gives me crappy estimates. I think I'll use those to design the big study. And that's really, I mean, you need to say these things out loud to really get into the spirit of the junk you're doing. Okay, okay. So most of the time where the delta comes from is, we're two places, a good place and a bad place. The good place it comes from is just discussions with clinicians. How big of a difference does it take for you to change your practice? And the second, the bad place it comes from is if I say this is 3%, I can do my study with 200 people. And if I say it's 2%, I'm going to need 800 people. I think the difference is 3%. Okay? Because I can't afford to do the study at 2%. Sorry. Okay, so anyway, the advantages of the equivalence approach, I can for a definitive statement, the new treatment is as good or better. Usually that's all a person wants. And I'll stop there and see if you have any questions because I'm getting ready to change the topic again. Okay. One-sided versus two-sided hypotheses. Okay? A one-sided assume that there's a new and old treatment, and the outcome is the proportion of people with events. Okay? And so basically a two-sided hypothesis is I assume that they're the same versus they're different. They're different can mean one is bigger than the other or the other is bigger than the other one. A one-sided hypothesis is that the new treatment is basically as equal to or bigger than the other versus it's less than. Okay. When does logic suggest we should use a one-sided hypothesis? Basically assume that the new treatment would not be used unless it's clearly better than the old. Okay. For example, assume the new treatment is surgery, old treatment is aspirin. If unless surgery is better than this, I don't really need to prove that medicine is better than surgery. I need to prove surgery is better than medicine. This is what happened in the asymptomatic carotid atherosclerosis study in academic childhood. Basically, at the beginning of the study, there was a 2020 story about how many needless deaths there were with endarterectomy. Surely surgery would not be used unless it can be proven to be better. So a one-sided hypothesis seems logical. Why would you use a one-sided hypothesis? Well, restricting the alternative allows more power, statistical power. Basically, assume that you want to see one group is 25% and be reduced to 20%. If I do this study with a two-sided hypothesis, I need to have 1,000 people. If I do it with a one-sided hypothesis, I only need to have 800 people. That's good. If I'm not interested in increased risk from surgery, then basically there's a 21% reduction in the number of people exposed. Okay, so when should you use a one-sided hypothesis? Never. Okay, you never do it. You never do it. Okay, go find one in the literature. Go find a paper that has a one-sided hypothesis in the literature. I dare you. Why not? Well, basically, you're providing valuable proof. A one-sided hypothesis is used. You cannot definitively say surgery was inferior. Okay, in that case, it's difficult to discourage the procedure unless it's proven harmful. Okay. Again, going back to my dear, by the way, I only work with surgeons. I love surgeons. Don't get me wrong. My closest colleagues are all surgeons, but they will not quit doing something unless you prove it's bad. Okay? Okay, one-sided hypothesis also smells bad. If you use one, it's really hard to get your data published. Any questions? By the way, there's a lot of opinion in my lectures in case you don't get the sense of that. So we've done the hypothesis. Now how about eligibility? Who do I put into my trial? Who do I want in my study? Okay. So basically, who do you want in your randomized trial? Well, there's a great need for homogeneity of my patient population. If my patient population has divergent or weird people in it, okay, it introduces noise in the system, okay? And it can really distort the outcomes of the majority. A lot of times in our study, we don't put stroke patients in our trials that are under 45 years old because people under 45 have strokes for fundamentally different reasons than people over 45. Could be drugs play a bigger role in young people, could be congenital conditions play a bigger role. It's not that atherosclerotic process that we're trying to fight, okay? So basically, the restriction of the population reduces noise and allows me to do my study in a smaller sample size. I don't know if y'all have ever heard of the Mr. Fit study. I won't go there. Let me skip that. I'm running behind schedule. But anyway, having a homogeneous population makes it so I can do it with a smaller number of people. Restricting the population to a homogeneous group is good. Alternatively, at the end of my study, I want to be able to generalize my results, okay? And basically, at the end of the study, it's important to only apply the findings to the people who are like the people in the study. Okay, if I do a study in men, I can't generalize those findings to women, okay? So basically, it's questionable to whether or not the studies can be generalized to those excluded from the study. And so basically, having a broad inclusion criteria that's welcoming to all is really a good thing. Okay? And obviously, these two things are perfectly and diametrically opposed. Okay? You want a homogeneous generalizable population is the answer to your question. Okay? So what's the correct answer? And the answer is there is no correct answer. Okay? It's a real judgment thing. And when you're reading trials, stop and think about who did they put in this study? Can this be generalized to my patients? Can I use these results? Yes, ma'am.
So when you were discussing the trial with the estrogen and the Women's Health Initiative in the beginning, they obviously had a problem when they were designing all of those observational studies, or I guess picking patients for those observational studies, or was there really nothing they could have done in the observational
Okay,
studies?
so the question is, going back to the estrogen trial, was that a matter of inclusion criteria and including the right kind of people in the study? It turns out that observational epi and in clinical trials always give the exact, precise, identical answer, always, as long as two things are true. Number one, you understand what all the risk factors are. And number two, you've measured them perfectly. Okay, the measuring them perfectly. So if I talk about socioeconomic status, how do I measure socioeconomic status? Well, by the way, the people doing the observational epi studies understood this. They tried to co-theory for socioeconomic status. They tried to fix this problem. How do we measure socioeconomic status? By far, the most common way is income and education. Okay, I have income and education. People make a lot of money. They're high socioeconomic status. I live in Talladega County, okay? There's a racetrack over there, and there are people who make a lot of money around that racetrack. And when you meet them, you're not impressed with the underlying gestalt of their high socioeconomic status. We all know people who make a lot of money, and they're not, this sounds horrible, but it's just hard to do a discussion with them. And income is not that measure. And the world is filled with educated derelicts. We all know very educated people And by the way, we also know very uneducated people who, are just my best friend. So we do a really crappy job of measuring socioeconomic status. By the way, if I'm offensive to anybody, accept my apology. But this is just, we do a bad job of measuring socioeconomic status. Unless we measure it perfectly, it opens the door for something called residual confounding, which means it's very much like cancer surgery. When you get some of the cancer, but not all of the cancer, I've adjusted for some of that confounding factor of socioeconomic status. I've done the best job I can, but there's still some stuff in there. Okay, and probably that's what happened to the hormone replacement is we just didn't measure socioeconomic status well enough. There are other theories why that came out that way too, but that's what I believe. Anyway, so who do you want in your trial? Clearly a balancing act. How weird are the weird patients? The results, can they be accepted for all subgroups, or is it really just one disease? Will subgroup analysis be required under any condition? You just have to use good judgment. Sorry. It's difficult to depend on subgroup analysis. My favorite subgroup analysis, my wife is very much into women's health and women's issues. And your NIH has very carefully made a statement about who, including women in studies. They say you must include an adequate number of women for a valid estimate. By the way, a very carefully worded sentence. Valid means unbiased. It doesn't mean precise or good or reliable. It just means that you get the right answer on average. Okay? It doesn't mean that you can tell if there's differences between men and women. It just means that whatever you estimate on average, if you did 10,000 studies and you averaged your male-female difference in your 10,000 studies, that answer would be correct. Okay? And I'll come back to subgroup analysis in a minute too. In fact, I'll come back to it very soon. Anyway, subgroup, it's common to approach special groups of people in subgroup analysis. Basically, in addition, men and women, is this treatment equally efficacious in men and women? In addition, it's natural and important clinical questions. There are subgroups in the population where the treatment was particularly efficacious, or there are places where it didn't work. These are natural questions, but require careful planning. You have to really plan these things out. And it sounds like I'm talking about confounding, and I thought confounding was not an issue, right? But that's not, this is effect modification, okay? Is the treatment effect, is it modified by what subgroup you're in? That's a different question than confounding. I wish I had more time to talk about that. So let me give you an example of subgroup analysis. And so basically, suppose I'm interested in and then I'll take a break after this, the relationship of diabetes and smoking to stroke among those with advanced atherosclerosis. And I use data from the ACAS study, that one that we talked about earlier. These are real data, real analysis, okay? And it was motivated, basically this is a randomized clinical trial of surgical management for carotid atherosclerosis. In that group, there were 800 people measured mathematically, and those are the people I used. Of those, 79% were normal and 21% were diabetic. And this is an example in this context of an analysis of one arm of a clinical trial. It's not really a clinical trial, but I'm just using this as an example to show you the kind of mischief and trouble you can get into. Okay? So basically, one of my heroines is Elizabeth Barrett Connor. If you don't know Elizabeth Barrett Connor, go do an email, a Medline on her, and you'll be impressed. She's a cool woman. And way back in 1984, she wrote a paper about the interaction between cigarette smoking and diabetes. And in that paper, what she noted was basically that cigarette smoking is a lot worse for you if you're diabetic. You don't want to, it's bad for you anyway. But if you have a patient who's a diabetic smoker, they're in deep trouble. Okay, and that was the motivation for this subgroup analysis that I did in ACAS. And so basically, it's, my question is there increased risk for cigarette smokers? Is it bigger among diabetics and non-diabetics? So my hypothesis is in the time until stroke, will it differ between diabetics and non-diabetics? And will that be greater for smokers than non-smokers? And so what I did was just a survival analysis, straightforward survival analysis. This is a real data now, real data. Okay. So here's my overall results from this. So this is looking at my diabetics and non-diabetics together. This is a Kaplan-Meier curve. I hope you're comfortable looking at these. And basically, the hazard ratio is 1.43. So diabetes has a 43% increased risk of having a stroke, but it's non-significant. P equals 0.14 in these studies. So what do I say? 43% increased risk. But I'm not really interested in that. I'm interested in the effect modification by smoking. Okay, so what happens if I look at it for non-smokers? So here are the data for non-smokers. Okay, and basically you can see there's only an 18% risk for non-smokers, and it's clearly non-significant. But among smokers, wow, look at that. I mean, I don't even need to do a statistical test. That's significant. Okay. Okay, and so I'll write a paper. There's a trend for diabetics to have higher stroke risk. It's primarily among smokers where the risk is nearly 3 times. Oops, I should have pointed that out. Let me back up. Look at this hazard ratio. Hazard ratio is nearly 3. That's amazing. Diabetes is really bad for you if you're a smoker. Okay, so basically what do I do is it's primarily driven by smokers, nearly 3 times the risk. These findings confirm my previous literature establishing an interaction between diabetes and smoking. It confirms my belief that diabetes is important, and it underscores the importance of smoking cessation in diabetics. Who thinks I can get that published? I can get that published right now. Right now. So what really happened? What really happened in this? Okay. So to begin with, I lied. Okay. That is not at all what happened. Okay. That's not at all what happened. At first, I was really interested in hypertension when I was preparing for this lecture, not diabetes. And so overall, hypertension was non-significant in predicting stroke risk. I was really surprised. And I looked for findings in other subgroups. And in fact, smoking, did not make a difference. While I was at it, I also looked at sex, race, bruise, alcohol intake, height, weight, BMI, a ton of other subgroups. Because when I started this, I wasn't really interested in smoking. I was just trying to find something that had effect modification. Okay? Nothing really excited me. I didn't want to write a paper about that stuff. That was not fun for me. Okay? And so it took me about 15 minutes to do that, by the way. The computers are the worst thing that ever happened to medical research. It's like giving chainsaws to children. I swear to God, you can do more damage. You can do more damage quicker with a computer than you can do good stuff. I mean, it's really bad. So, the next thing I did was analysis of cigarette smoking. Overall, it was non-significant. Nothing really excited me. I did the same 44 analyses there too. It took me another 15 minutes to do that. So now I've invested a whole 30 minutes into this paper. Okay. Finally, I turned to diabetes, and overall it was non-significant, as we showed. I saw this association in smokers, and then I remembered Elizabeth's paper. At that point, I said, oh, Elizabeth Barrett kind of gave a talk about that one time, and I was in the audience with Elizabeth, and she said it was bad for you to be a diabetic smoker. That was not a priori in what I presented to you. That was a story. I found this, I remembered this paper after I was doing this. I ignored the significant other findings, by the way. Ever drinkers were also at higher risk, okay? Previous contralateral endarterectomy, bruise were at higher risk. Whites wasn't significant in blacks. History of hypertension was in fact not the middle tertile of weight. I'd love to write a paper explaining why skinny people and fat people aren't at risk, but average weight people are. But the mechanisms can be hypothesized for some of these strokes. Basically, overall, I did more than 100 analyses. Okay? It took me about 40 minutes to do this. I really care about y'all. I really worked hard on this lecture. It took 40 minutes to do that. Okay. I reported only things that confirmed my biases and only things that was my sense of my literature. And was I cheating? And if so, how bad did I cheat? And the answer is I cheated bad. I mean, so what this graph shows is, and this is one of the ones I want you to keep in your mind, this is the number of comparisons I'm making here. And this is the probability that I find at least one significant finding. Yeah, I find at least one significant finding. So by the time I get to 5 tests, I'm at a 20% chance of finding something. Okay. By the time I get to 30 tests, I'm at an 80% chance of finding at least one significant finding. 30 tests, by the way, is not a lot. If you think about these papers that have that forest plot at the end and it shows all the different subgroups and everything, very frequently that 20 or 30 different factors are looked at there. Okay. You know, I flagrantly cheated. And by the way, intentionally flagrantly cheated. And the results remain very publishable, but likely worthless. Okay. Almost through the subgroup analysis. So if subgroup analysis is natural and important, how do you want to do it? Well, to begin with, if you don't know what Hill's criteria are, write it down, go find Hill's criteria for causation. The idea is when do we really believe these findings? And there are 7 criteria. One of them is a plausible association. You know, something that underpins the association that you're seeing, the mechanism for it to work. And it's hard to believe things that don't have a mechanism. Don't have a mechanism. By the way, that is the weakest of the, in my opinion, the weakest of the criteria. One of my favorite stories is an ACAS, that study that I've been talking about some. At the end of the study, we found that endarterectomy was a lot more efficacious in men than women. Totally unexpected finding at that point. Did not hypothesize it at all. I love Jim Toole, my mentor. He sometimes said things in awkward ways. He got on a podium and said, that's because women have delicate arteries. Okay, delicate arteries. By that, he meant their arteries were smaller and harder to operate on. And that is the reason why the surgery is not efficacious in women, is they have delicate arteries. And I was talking to him after this lecture, and I said, Jim, what would you have said if it had been efficacious in women but not men? He said, then I would have said, men have more advanced atherosclerosis, and surgery on men is much more dangerous than it is in women. You know, once you see the association, playing the game of why is it there is remarkably easy. Finding the literature to support it is remarkably easy. Don't believe it. You know, so what do? I want to limit my analysis and keep track of what I've done, and I want to report what I've done. I want to report in my paper how many analyses I actually did. I want to, if you're a consumer of the subgroup analysis, if you're reading the paper, watch out. Beware of this. Be careful. Okay? You know, the most logical and clinically important subgroup analysis may well only be one of 10 that was done, and look for the authors to point out that what they did was an a priori analysis. And so this is the last part of effect modification. This is from the Evascott trial. And so these are the different subgroups, which shown here is the statin was better, the statin was worse. What is your interpretation? What do you say? Well, look at that. My goodness. It doesn't work in women. Okay, it doesn't work in women. You know, should we not treat women? Is it less effective in diabetics? Look at diabetics here. Looks like in diabetics, it's, you know, it's sort of different. You know, so basically there are differences in 18 a priori strata with no significant interactions. Okay, so basically what this is really demonstrating is a homogeneity of treatment, not heterogeneity of treatment. And does this figure help you understand the findings? And it should. When you look at that, you should say, wow, look, what a consistent effect across all the different subgroups. Hopefully is what you'd say. And with that, I think we should all stand up and move around. Take a break for about 5 minutes, if that's okay with y'all. Wash your hands, as my mother taught me to say. Is this okay? Is it interesting?
Yeah. So, if you have a trial, right, and you find an effect and it's positive, When did they decide I'm going to stop the trial because the effect is so positive?
I'm going to talk about that.
Oh, good. Okay. I've always wondered about that.
So what that is right here. This is work.
It doesn't want to work.
That is interim analysis versus adaptive design.
Okay.
This does.
I'm excited. I've always wondered about that.
So I talk about.
Cool.
I'm not sure about
It's good. I really miss that a lot.
Yeah, that is true.
It's the
not
worst.
consistent.
It hides my own center, doesn't it? No, but the other stuff's great. The amount of
Other than their EMR,
Is this confusing?
Well, yeah, no, following one thing, you mentioned a priori and then another phrase that sounded like
A priori.
a priori. Well, I'm not familiar with
A priori means that before I looked at the data, I made up my mind I wanted to ask this question.
Okay.
I did not look at the data and find the difference and then say, ooh, this is interesting. I wonder why.
Gotcha.
And so a priori means that
You thought about
I thought about it first. Because once you look at there's a name, statistical name for looking at the data and then making up your hypothesis. That's called cheating.
Yeah.
And don't cheat.
So in that case, had diabetes and smoking been cited as an a priori variable, you might have included.
I might have been. Well, I mean, if I really had read Elizabeth's paper, and thought, oh my goodness, I wonder if that's true in these data, that would have been a valid analysis. And chances are we would have tried to publish.
Right.
I mean, I know I can get that published. I don't think I can get that
I'm a diabetologist, I believe that.
published. I mean, yeah, confirm true.
Yeah. Okay.
Or anything because you didn't cut a hypothesis.
Oh, I cooked those data big time. That was an exercise in cooking data. I exercise those data until they scream stop. I mean,
But now you can never use the data?
I can never write that. Well, I can take that finding. By the way, a lot of times you do find things that are unexpected. So what do you do then? You know, once I see this, what I do is I go try to find somebody else's data that's similar to my data and see if it's true there. There I'm doing it in an a priori setting. In other words, I can go find another study and see if the same thing is true in that study. Then that's a pure examination. It's not contaminated. And for that reason, it's really good in papers when you do find things you don't expect to publish the fact that you found this. They need to be published in the spirit of saying, when I was poking around in the data, look what I found. I hope you see this in your data too. Please check and let me know. You know, that which is different than snooping around in the data. There's almost a perfect correlation between the street temperature of New York City and infant mortality. Perfect correlation. I don't think that's causal. I think that was found by somebody doing a great big correlation matrix and picking out the big number. Because if I look at 20 things, you know, that's above 50% chance I'm going to find something significant. Oh. A symbolic correlation. Okay. Okay, I'd like to keep going because I would like to get down into the analytic issues at least. So anyway, the next point is looking at efficacy versus safety, something that at least I was always confused about when I was getting into this business. And so basically, I really wish I had more time to talk about this. This is one of these things that seems so simple, but when you open up Pandora's box, it gets really confusing. Okay, So basically, trials are designed to assess differences in the primary outcome. Consider, the hypothetical case of a treatment approach that improves the modified Rankin score of 80% of the patients by 90 days. That's my primary outcome. That is, it's a very efficacious treatment. Really works. Unfortunately, the other 20% of the people had a hemorrhage on day 1 and died. Okay? The treatment's efficacious. Works. Okay, my primary outcome is significantly better. Would you recommend that treatment? And obviously not. I mean, you're not going to kill people. Generally, unsafe treatments are not employed even if they're quite efficacious. And biopsy is the example of that. And so how about efficacy and safety? So clinical trials are focused on establishing treatment efficacy. Safety is important, so why not have that as one of the primary goals of a trial? Why don't we have safety elevated up to the level of efficacy? And the issue is that while you're pretty sure about what you want a drug to do, at the beginning of a trial, generally, you're not completely sure about what you don't want it to do or what it can do. Okay, you only get that question sort of at the end of the study. You go, oh my God, look at all those heart attacks. I didn't think that was going to happen. Okay, and so basically, that means within trials, for efficacy, I have clearly defined and well-specified goals. I know exactly what I'm going to do for assessing efficacy. Okay, for safety, it basically comes down to a gut check. Is this something that I feel good about treating my patients with? Because you can't say something like if you have 20 more, if the ratio of serious adverse events is 20% higher, I'm going to quit. Well, I mean, if it's 5% higher and all the adverse events are death, you're going to quit too. Okay? In other words, that these things don't get equal weighting in your thinking. Okay? More later if we have time, but this is one of the reasons that you have a data and safety monitoring board. That's the last thing on the outline, and about 3/4 of the times I don't get to that part of this lecture. Yes, ma'am.
Just a quick question. I do clinical trials in pregnant women. Do you think it ever would make sense to have the efficacy and the safety outcomes be equally important?
The question was, if you do have efficacy and safety outcomes be equally important, I cannot imagine a situation where that would be the case. I mean, I think that I have been part of trials where it was fairly clearly understood what the main safety concern was. My example in my world of that is hemorrhagic stroke following TPA treatment. I mean, the thing that scares everybody is I don't want these people to hemorrhage, okay? The way that you can get around that is to bring that safety concern in and have it be part of the efficacy. And so what you can do is have some kind of function of both of those so that I want to improve while minimizing, so you can bring it on the other side. But most things that are safety concerns, I mean, in CREST, the first 8 people who died, damn it, were all in one group. Can you imagine being on the safety monitoring board and 8 out of 8 people had one treatment? Thank goodness, at the end of the trial, there was absolutely no difference. Deaths were perfectly equal. I mean, strange and unusual things happened. We never anticipated a difference in death. I mean, that was completely out of the blue. And it ended up evening out by the end of the trial. But boy, there were a lot of telephone calls by the DSMB. I mean, we had like weekly calls. Everybody died this week. It was really serious. Anyway, so basically that's all I'm going to say about efficacy and safety. Masking, important part, which is also called blinding. Okay, and so basically the concept is that we all know what the outcome of trials is going to be, even though we're at equipoise. This is it does not change the fact that we all have these preconceived notion of what the right thing to do is. By the way, you wouldn't be doing the trial if you didn't have some feeling that the new treatment was better. Some feeling, and there are great examples of times that it wasn't better. And the best example of that is the alpha-ketophrol beta-carotene study, where basically what they found were people given, in 1960s, amazing thing, people actually believed that if you loaded somebody up with beta carotene, they could get away with smoking. If I gave them enough beta carotene, you could protect people from the harmful effects of smoking. Amazing thing to say today, but that's what people believe. So they did a trial, loaded people up with beta carotene, and alpha-ketopherol too, but beta carotene, and found out that wasn't true. But not only was that not true, but the people on the beta carotene had more cancer than less cancer. They also had more car wrecks, they had more heart attacks, they had more strokes. Every outcome was worse. And that leads to one of my favorite cartoons ever. Well, you know, I'm sorry, both people and placebo, both patients and investigators assume the active treatment is better than the placebo. And my favorite cartoon, Hiram Dudson, member placebo group. But, physicians want to help people. You wouldn't be doing this if you didn't want to help people. And so the evaluator, if the evaluator knows the assignment, then they can be more intensely looking for worse outcomes in the people who don't get it. Are you sure you're okay? Sure you're okay? And so basically, so how do you avoid that? You know, well, the first thing you do is you have the same protocol in both arms. Unequal evaluations can provide opportunities to discover events. You want to be very careful to treat both groups the same. You know, examples of not being able to do that is if I'm doing caloric restriction, then I have one group come in for weekly classes and the other group doesn't. Well, when they come in for weekly classes, if somebody comes in with, you know, paralyzed, you say, oh, did you have a stroke? And that person's not found in the other group. Okay. Surgical you know, may require follow-up visits. You know, every visit is an opportunity to discover an event, so you want them treated the same. You know, a patient can't have a fever if you don't take the temperature. Okay. Masking to protect randomization. Both biases can be avoided if neither the patient nor the physician knows the assignment. That implies that, that the follow-up schedule is identical. The old term is blinding, the new term is masking, if you want to be really cool. It really came out of the Eye Institute. They really don't like to do studies of blinded people at the Eye Institute. They don't consider that a good term. If you are doing something with Eye Institute, you want to avoid the blinded word. They actually object to it. You know, double mask is neither the participant nor the investigator knows the treatment. Single mask is usually the patient doesn't know the treatment. Challenges to masking. It's not always possible. You know, residual surgical wounds, you know, if you're doing endarterectomy versus treatment, everybody in one group walks in with a big, you know, scar on their neck. Responses to masking, cycling and HRT replacement. I was part of the PEPI trial, and one of the treatments was unopposed estrogen. You know, it's going to be fairly clear who's on unopposed estrogen. Okay. Serum or other assays. You know, participation in the treatment. You know, if there's diet or exercise in one group. And the common approach, if you cannot do masking, is to at least mask the rater, separate the rating of the outcome from the treatment and have separate raters. And the term for that is a prospective randomized observer blinded evaluation design, a probe design. And so, for example, in CREST, the endpoints, the surgical trial stenting versus endarterectomy, we bring back every suspected stroke. We have a team go through and mark and remove any evidence of what treatment that people were on. They're reviewed by a committee that doesn't have any idea which group they were in. It's incredibly expensive, incredibly time consuming, and really worth it. You can also move towards a more objective, whoops. How do I go backwards? A more objective measure. I don't need to blind deaths. If death is my outcome, I can adjudicate deaths. I mean, you know, it's going to be hard to be biased in my assessment of it. So the more objective your outcome is, the less important masking is. You ought to think about that in design trials. Have an independent observer who doesn't know the treatment. That's the probe idea. So moving on to analytic issues, pushing through, hopefully finishing up on time. First of all, is intention to treat. I'm sure all of you are familiar with this, but just in case you haven't heard this part of a clinical trial, okay, basically assume we have 100 patients in 2 groups, surgery versus stent. Of those in the surgery group, 10 patients announced the only reason they were in the trial was to get the stent and now demand the stent after randomization. This happened to me. This didn't sort of happen to me, this happened to me. The only way that you could get a carotid stent was to be in our trial. And people agreed to be in the trial because they wanted the stent. Okay. Do we compare those getting the stent to those not getting the stent? Or do we do those assigned to the stent versus those assigned to surgery? And wouldn't be called intention to treat if we didn't do B? So on the surface, it makes a lot of sense to analyze those people who received the treatment. There's a name for that. It's called treatment received analysis. These patients were where the treatment, by the way, explain to a surgeon why somebody assigned to surgery who got the other treatment and they had a stroke during the other treatment counts against the surgery. I mean, if you don't think that this can be contentious, I mean, that is actually a discussion that's very, very interesting to have with the surgeon, that the guy putting in the stent caused the stroke and it's going to count against you, okay? However, the whole idea of randomization, the complete idea of randomization is the 2 groups are equal. I've removed confounding, potential confounding. And it's possible the chance of changing treatment is associated with some trait. My real world example is young, well-educated, aggressive people wanted the stent. So those 10 people who went to the other group weren't random people. They were the young, smart, highly educated people who changed groups. Okay. Those receiving the stents are now younger and they are better educated than those not receiving stents. And at the end of the study, how do you know that any observed differences were not attributable to age, or more importantly, to some unobserved risk factor that you don't know about? Okay? And so the answer is analyzing the treatment received loses randomization, doesn't protect randomization. Randomization is the key to all of this. Okay? Any questions about that? Yes, ma'am.
Is there some percentage beyond which you can't do that?
The question is there some percentage beyond which you can't do that? Again, it's all just subjective. I mean, it's so funny. In CREST, the study that I'm doing now, we're doing 2,000 patients. And at the end of the day, we're anticipating about 44 stroke events. And so that 44 people are going to change how medicine is practiced. And so basically in a study like that, you really have to protect, because having 3 people come over and having an event, heck, that's about 10% of the events, or because of crossovers, okay? So a lot of it depends on the context of the study. If it's a trial that has more events, you can tolerate more crossovers. We work very, very hard to not let crossovers happen because we're anticipating very, very few events. And if 1 or 2 people come over and have an event, nobody's going to believe our study.
So then you just push them out of the study? I mean, instead
Nope, you lose randomization because I'd be excluding all the young, well-educated people. Okay, once randomized, always analyzed.
Unless they drop out.
Unless they drop out, and even then you try to keep them in with techniques that I wish I had more time to talk about, called multiple imputation and inverse probability weighting and other things that we're not going to get to in a one-day lecture. But you keep them in even then. Even if they drop out, you keep them in. Unless they say the magic words, I rescind my informed consent. If somebody says that's the only way that you get out of the analysis, is to say, you may not use my data, because you have to respect that. If somebody says you may not use my data, the answer is yes, ma'am, or yes, sir, and you dump the data. But you tend not to tell people they can say that, and people don't randomly say that. But sometimes you'll get an MD in your study and they'll say that kind of stuff. And you have to say yes. So basically, statistical power, an important part of designing a trial. What in the heck is statistical power? So we spend a lot of time doing trials in this column under the the assumption that there is no difference in the two treatments. Living in that column, if I assume there's no difference and I see the test concludes there's no evidence of a difference, then basically I make a correct decision. I said there was no difference, there was no evidence of a difference. Yeah, I got it right. Okay, if the test concludes there is evidence of a difference, then I made an incorrect decision, I lose, and I reject the null hypothesis, and that's a type 1 error. In other words, I've said there is a difference when in fact there's not. Okay, I've said there's a difference when in fact there's not. I've made a mistake. I'm going to say this new drug works. It's going to be cranked up and put into the practice, and I was just wrong. By the way, that happens. I've done a lot of trials. 5% of my trials, I've done that. Okay, by definition. Okay. We don't spend as much time over here, and this is in the column where you assume there is a difference. So when you mess with this column is when you're designing your study. When you're designing your study. So now I'm going to assume that there's a difference. Okay? And if I assume there's a difference and I conclude there's no evidence of a difference, I've made an incorrect decision. The drug really worked. In truth, it's an efficacious drug. But I'm going to say, I don't see anything going on. And by the way, that's called, you know, I love statisticians. We took a long time to come up with these terms. If that's type 1 error, this has to be type 2 error. Okay. Okay, anyway, so, and that's the beta. So basically, y'all are used to thinking about this. You probably haven't thought about this quite as much. Okay. In other words, both of these are mistakes. Type 1 error is when I say there's a difference when there's not really one, and that's bad. Type 2 error is when I say there's no difference, or I don't see a difference, when there really was one. That's equally bad. Okay. I have an efficacious drug that will never be tested again, ever, because once you do a trial and say that there's no difference, randomizing people to it becomes unethical really quickly. So you just killed an efficacious drug. Okay? That's called a type 2 error. Alternatively, if there really is a difference and I say there's a difference, I've made a correct decision, I win, and that's, if I don't do this, I have to do this. So 1 minus that is the statistical power. Okay? Now, alpha here, for the funniest reason that you've probably never heard, is always 0.05. Okay. Hadn't y'all always wondered where that came from? It's the interesting thing. Where did 0.05 come from? It turns out it came from a guy named R.A. Fisher, who basically is the Michael Jordan of statisticians. He is the dude. He is the dude. I would say half of what we do as statisticians came out of this one guy's head. And back when they were working out hypothesis testing, he was at a meeting. By the way, never got his doctorate. And so somebody came to the microphone and said, Mr. Fisher, how much evidence does it take for you to reject the null hypothesis? And his answer included the phrase, gee, I don't know, usually about 1 in 20 does it for me. And out of that, 0.05 happened. Okay, just one guy's statement from I don't know if there were microphones then, but from the podium anyway. Okay, let me have you question alpha for a minute. Question alpha for a minute. I'm doing 2 trials. First trial is I'm interested in does orange juice prevent colon polyps? Okay, and I have a p-value of 0.09. As a clinician, would you recommend drinking orange juice?
Is the person a diabetic?
So there's potential safety issues. I would probably recommend it. I sort of like orange juice. You know, other than if you're diabetic, it's not that many bad things can happen to you. You can, you know, it's sort of medium evidence that it's beneficial. Okay? Shoot, let's have people drink orange juice. On the other hand, I have this other trial where I'm going to take infants and I'm going to split open their chest at birth. I'm going to massage their heart for some reason I can't make up standing up here right now, but it makes them better at 5 years. P is 0.04. Would you recommend that treatment? And the answer is, wow, there's a 4% chance I'm wrong, and I'm getting ready to inflict horrible interventions on a large group of people. I'm not willing to do that with a 4% chance that I'm wrong. That would be the rational thing to say anyway. I want to be a whole lot more sure than 0.05. A whole lot more sure than 0.05. And so basically, I think we're moving as a society away from 0.05. I hope we move away more rapidly. I hope we can make intelligent clinical decisions about what we do and don't do based on the full scope of the evidence and not a silly dichotomization of alpha level. For other similar stupid reasons, power is normally 90% or 80%. There's 80% chance, 80% power. When I say a study has 80% power, that's saying if the difference really exists, I have an 80% chance of seeing it. Let me say that again because that's an important one. If the drug really works, I have an 80% chance of seeing it. When I'm working with clinicians and they want to do an 80% trial, I make them say this sentence: I'm getting ready to spend the next 5 years of my life working on a project where if the drug really works, there's a 1 in 5 chance I won't know it. Okay, that's what 80% power really means. Because your life is a series of 5-year periods, and how you allocate your 5-year periods in the NIH grant funding world is a strategic decision. And doing something that has 80% power might be a questionable way for you to spend one of your 5-year periods. Okay. By the way, 90% power still means there's a 1 in 10 chance I won't see it, which ain't a lot better. But that's what power is. And so basically, type 1 error, chance of saying the null hypothesis is false when it's really true. You don't need to decide. You need to decide how much risk to take. That's the alpha. Normally 5% chance. In fact, write a grant and have something besides 5% in it and see how it fares in review. You don't really have a choice, unfortunately. Type 2 error is failing to say that the null hypothesis is false when it really is false. You need to decide how much risk you want to take, normally 0.1 or 0.2. That's either 90% or 80% power. So if the null hypothesis is false, you want to say it's false, and beta is the chance of making this mistake. So 1 minus beta must be the chance of making a correct decision. And 1 minus beta is the statistical power. And I want to stop and make sure you're all cool with this idea about what I'm talking about. This is one of those points I'd really like you to take on. Okay. So how do we do this? The easy part of deciding statistical power are the formulas and such, and there are 5 intertangled numbers. The first one is the level of evidence I want to have to declare significance, and that's my alpha. Okay. The second one is the likelihood of seeing a significant difference if one really exists, beta. Okay. How big the difference is between the treatments. In statistics and in life, big things are easier to see than little things, okay? The bigger the difference, the easier it is to see, okay? That's the delta, okay? The sample size, okay? The only reason in your life that you'll ever do a study with 200 people instead of 100 people is to buy power. But basically, sample size is largely the currency of power. Okay? That's N. Okay, the noise are other parameters in the system. This is, again, not rocket science. If you're in a noisy system and a lot of stuff is happening, if you do crappy measures on your patients, it's hard to see a difference. The reason you take blood pressures twice and average them is to remove noise. Okay, because you can see differences if you take it twice and average them better than if you take it once. Okay, that's signal. Okay, so basically, if I know any 4 of those, I can calculate the 5th. Okay, if I know any 4 of those, I can calculate the 5th. So basically, if I don't know the likelihood of seeing a result if one really exists, if I don't know beta, then what I'm doing is called a power calculation. Okay, if I don't know the delta, then what I'm doing is called a detectable difference calculation. Okay. If I don't know the N, then what I'm doing is called a sample size calculation. Okay. But basically, it's the same formula, and all I'm doing is, I could write out the formula if you're interested. We can do that after. The thing's really easy formula. You never do it by hand. You always get a computer to do it, so you don't need the formula. But basically, it's the same formula, and all you're doing is solving for the one unknown. Okay. Somebody earlier asked about interim analysis. Okay, interim analysis. The idea of interim analysis is, suppose that halfway through the trial, you really see these two curves and they're really diverging. Well, big things are happening. Okay. I'll argue that you're both clinically and financially bound to do a study with as few people as possible. Okay. So there are two ways to think about reducing the number of patients. Okay. Well, let me back up one step. I'll do it after the slide. There are two ways to reduce the number of patients. The first is called group sequential trials. Okay. And in group sequential trials, I plan a big study and I try to stop it early. Okay. So basically, there are common approaches. They're called O'Brien-Fleming, Landametz, Pocock, Peto stopping rules. All of those are summarized into, I do love this term. It's called an alpha spending function. An alpha spending function. So at the beginning of the study, I have 0.05 of my alpha in my hand. And I want to look early to see if there's a difference. Okay? I want to look early to see if there's a difference. Well, the more times I look, the more likely I am to find something. I can't frivolously look at the data to see if there are differences. I need to plan very carefully how to look at the data. And these are all the rules for carefully deciding halfway through the study, I'm going to look one time. Okay. And what I have to decide is how much of my 0.05 do I want to spend for that peak at the data. Okay. And so the difference between these different stopping rules are simply whether I want to spend a lot early and hopefully gamble and get my study stopped as early as I can, or whether I want to hoard that 0.05 so at the end I have as much of the 0.05 as I possibly could have. Because the worst thing that I can imagine happening, the paper I hope to never write, is something where I've spent, let's say I've spent 0.002. So my final look is going to be at 0.048 instead of at 0.005. Let's say that you come up with a p-value of 0.049. That puts you, because I've already spent 0.002, that puts me in the position of writing a paper that says these two treatments are not statistically different. P equals 0.048. And at that point, I've confused every naive clinician who's not taking the time to take classes like this. They all think I'm nuts. You know, it would be a horrible experience. So I want to hoard as much of that 0.05 as I can for my final peak. Okay, and only if there's a huge difference do I want to. So basically, the whole idea of looking early and trying to stop early is a subjective decision of how much risk do I want to take at the end of the study and how certain I am that I want to do it. So basically, the most common of these are the O'Brien-Flemings. It's the most common one used. It's a hoarding function. You end up with like 0.048, 0.049 at the end. Unless the differences are massive, you will never know it. Okay, the most liberal is the Peto. Okay, basically what that says is I'm going to look 5 times, 0.01, 0.01, 0.01, 0.01, 0.01. Okay, which means anything bigger than 0.01, 0.03, non-significant at the end of the study. Okay. Okay, so that's the group sequential trials. What have I done with the clicker? Has to be in my pocket. Not in that pocket. It's in that pocket. Okay. There's by far the most widely accepted and valid approaches. But there are 2 ways to think about reducing it. The other one is the adaptive trial design. Hot topic, hot topic. You will sound really smart if you do an adaptive design. People will think you're cool, okay? And the adaptive trial is to start with a small study and see if it needs to be made bigger, okay? And basically, it's a rapidly evolving area of statistical research. Adaptive design is really an ill-defined term. Like, I'd love somebody to tell me what bioinformatics means. You know, I've never figured that one out. Adaptive approaches fall into two broad domains. Okay. The first domain is adjustments in my sample size for estimation of nuisance parameters. Okay, a nuisance parameter is not the treatment difference, but on average about how many people are going to have a stroke. Because if I'm designing a study that expects 10% of the people to have a stroke, I don't need as many people as if it's 2%. Okay, but if I get my study started, I'm halfway through, and 2% of my people are having strokes, I'm in deep doo-doo. I need a bigger study, okay? And then adjustments on interim estimates of the treatment effect. And so there, what you do is I'm halfway through the study, I thought there would be a 2% difference in treatment. Halfway through, oh my God, it's only 1%, I need a bigger study, okay? Much more controversial, much more suspect, much more likely to get you in trouble, okay? And so the estimation of nuisance parameters is well accepted, and the jury's probably still out on whether you can do adaptive trials on estimating treatment effects. There's a great, if you're interested in this, there's a great FDA position paper on it. I'll be glad to share. So which approach is better? You know, is it better to plan a large study and see if I can make it smaller, or small study and see if it needs to be bigger? Both seem reasonable. So let's look at three criteria. Which one provides the smaller sample size on average, which is logistically or politically easy to do, and other criteria? So in terms of which one is the smallest sample size, this one's easy. To cite us in Meta in 2000, any adaptive test, no matter how it can be attained, can always be improved upon by a likelihood ratio test that utilizes the same acceptance and rejection regions as the adaptive test. You don't understand that. But statisticians almost never get to say the word always or never. We don't get to use those words. No matter what you do in an adaptive trial, there's always a group sequential trial that is as good or better than your adaptive trial. Always, 100%. That's what that says. So adaptive trials, smaller sample size on average. Regardless of how the adaptive design is obtained, there's always a group sequential trial that is as good or better, okay? Which is logistically or easier to implement. You know, it may depend on who you are. You know, if you are a pharma, suppose you have a compound where there's very little experience, you need to get it to market as quickly as possible. An adaptive design may be a great way to get something started, start collecting my data, get out in the field. You know, if it looks promising, I've got infinite resources. I can dump more resources in and make a bigger trial. But, you know, I'm a little bit confused. You know, why would one start a program that's likely to cost millions of dollars where you don't have a lot of experience, but it can be done? Let's assume that you're an NIH researcher, which most of you will be. You know, imagine saying to the NIH, I'm not really sure how big this study is going to be. Why don't you give me some money and I'll get started, and then I'll tell you how much money I really need. You know, that is going to be hard to get funded, okay? In the NIH world, stopping early is a big win, but it might not be a lot, you know, the exception to the rule, which we probably should be doing adaptive trials, is no study always finishes on time. And so when you're doing your grant renewal, it'd be a great time to do an adaptive assessment of, do I need to make my study bigger? And then the other points are the interests in saving the study. You know, should there be boundaries on design? Suppose that I was originally looking for a clinically significant difference of 25%, and it required 200 people. Halfway through the trial and interim analysis, you know, said it could be, you know, could be reduced to 10, with the estimate of the hazard was reduced to 10%. You know, if this is a correct estimate of the hazard ratio, you only have 22% hazard, power. In other words, if halfway through the trial I'm seeing smaller differences, do I want to make a bigger trial? And so is it right to save the study by increasing the sample size? Okay. But then that raises the question of why are you changing your mind about what a clinically significant difference was halfway through the trial? That's why it's controversial. I did a bad job on that slide, but I'm time urgent now. But anyway, other issues, the impact on study blinding. In a group sequential trial, if I'm going along, everybody knows when you do your interim analysis, the world knows, everybody sits and waits for you. And I finish and you don't say anything, so the study keeps going. It's sort of expected to keep going. So nobody really feels like they've been informed by the study keeping on going. Alternatively, let's do an adaptive design. So I blind the investigators and the participants, and I perform an initial analysis and announce that I need to double my sample size. There's a lot of information that I've just given out about what's going on. I've largely unblinded the study by that statement, because that says it looks promising, but my nuisance parameters were not what I thought they were, and we need to make the study bigger. So basically, remember here we focused on group sequential versus adaptive designs, and using interim estimates of the treatment effect. If not, adaptive trials focusing on nuisance parameters are still a good thing to do. Criteria for making adaptive group sequential decision, the reduced sample size of power provides greater power. Group sequential trials are uniformly superior. Logistics of getting it done, group sequential trials seem to have fewer issues. Other issues, it's not clear that saving studies makes a lot of sense, and maintaining blinding might be a hard thing to do. In case you don't get the sense of it, I'm not a big fan of adaptive design. I'm a big fan of group sequential designs, at least in my world. It does make you feel really smart, though. When you go and say, I'm doing an adaptive design, everybody just thinks you're cool. Everybody thinks you're cool. I am going to quit because there's only 10 minutes left. Well, let me do Bayesian and then I'll quit, because Bayesian is one of the hot topics. And so anyway, from a 30,000 foot view, this is just a raging debate in biostat. Frequentists, what they do is they consider the true treatment difference to exist in nature. There is a % difference that this is either better or worse, the new treatment than the old. It exists. My job is to guess that %. Okay? Bayesians think of the difference as being a distribution. It's not a number, but a distribution of numbers. And we already know a lot about that distribution from previous studies. I don't come into these studies, blindly, not knowing what I'm doing. I already know, I have a good feeling for the likelihood that this new treatment is better. Okay, and our goal of the study is to update what we know about the study, about the treatment difference, using the information from the previous information and updating it with the study. That's the whole idea of Bayesian analysis. And I don't need to say, if there's a lot of substantial prior information, it can really powerfully inform your study. It's a huge win. However, if there's not a lot of prior information, then there's very little gain, and this is really important. Some argue that Bayesian analysis allows adaptive designs to be implemented. This is true, but it's also true that they can be implemented under frequentists. There's a guy named Barry who's made adaptive Bayesian design one word. It's really two words. You're the adaptive or you're Bayesian. You'd be Bayesian and Bayesian and adaptive. Some argue that Bayesian analysis answers the logical question. And it's actually easier to argue both approaches. So if there's so the true gain and the real opportunity to seize the prior information and reduce the sample size. I can use that prior information, reduce my sample size. Is this something we really want to do? Okay, so 100% agree Bayesian analysis have the advantage of optimally using prior information from previous studies to inform phase 3 trials. This allows a smooth transition from phase 2 to phase 3. I can do my phase 2 and just sort of keep going using that phase 2 information to inform my phase 3 information. What kind of fool would not want to use all the information that is available? Me, okay? I am the fool that does not believe in Bayesian trials, okay? Gee, why not? Well, there are problems about what we know. You know, we have to use Bayesian analysis, we have to agree about what we know. And we have to know exactly the right amount. And we may be systematically wrong about what we know. So those are the three problems that I have. So let's stop and think, do we really want to use that information anyway? So here's problem one. We have to agree about what we know. Let me back up. So basically, Bayesian analysis 101 is I have this prior distribution of a treatment effect. I'm going to do my study. I'm going to tighten up what I know about my effect. I'm going to shrink my uncertainty. So basically, that's cool. But let's say I have a different prior and I do the same experiment. And now I have a different posterior. So this posterior is different than that posterior, even though I've done exactly the same experiment. Why are they different? Well, I've disagreed about what the prior was. Okay? And so basically, you know, what do academics do? Well, we write papers, we care for patients, we teach students. What do we do poorly? Agree about what we know. Okay, getting people to agree about what you know is not a trivial fact. Should I include animal data to inform my prior? You know, it's really a hard decision. So think about the chaos that would result from a $50 million study after which people disagree about what it even means. Second problem, we have to know just the right amount. Okay? Remember that we only gain from Bayesian analysis if we know something. If we don't know something, we gain nothing. Okay? So if we know nothing, we gain nothing. Also remember that randomization is based on equipoise. If we know too much, are we at equipoise? You know, can you imagine, giving somebody informed percent? We're only about 80% sure that you'll benefit from the new treatment. However, to remove our uncertainty, we would like to assign you at random to treatments. I think you need to do something like that for Bayesian analysis. And where do you draw the line? If you're 80% sure that new treatment is better from your prior, are you comfortable with randomization? I would not be. 60%, I might be comfortable. 60-40, maybe I could still randomize people. So you have to know the right amount. You can't know too much, and the less you know, the more ethical it is. But the less you know, the less you gain. Okay? You know, what are you going to do at randomization? The third problem I have with Bayesian trials is we may be systematically wrong about what we know. You know, you can pretend that the current study is the only study that you're doing. Okay, it's the only compound that you're assessing. You can also pretend that I hope to arrive to this lecture on the unicorn. You know, it is not true. This is not the only compound that is being tested. In fact, from 30,000 feet, there are hundreds or thousands of compounds that are introduced to assess efficacy. And the only ones that move forward to phase 2 and phase 3 are the compounds that look promising. Okay? There are two reasons why a compound could look promising. The first reason is it's promising. And the second reason is through a random process, it inappropriately appears to work, which means that all information that phase 3 trials are based on are systematically biased and overestimate treatment effects. If you look at all phase 3 trials, they always show a smaller effect than the phase 2 trials. Okay, and the reason for that is the phase 2 trials are biased. They're going to overestimate the treatment effect. If you introduce a biased prior in, you're going to get a biased posterior. Okay? So conditional on making it to phase 3, phase 2 studies are biased. The phase 2 is biased, and incorporating that information will bias the phase 3. And then the final point is, do we really want to use that prior information anyway? The knee jerk is that we want to use all the information available. Suppose that we're getting ready to do a pivotal clinical trial. Doesn't it make sense to wipe the board clean if I'm doing a pivotal trial and have all of my decisions based on that trial? I mean, that's the whole idea of being a pivotal trial. You know, do we really, do we really understand the priors? You know, some could argue that some of the previous scientists had a program. Understanding of the information is, is really there. Is it really a bad decision to have an independent assessment? And I'll argue it's really a great idea to have an independent assessment. And with that, I will quit this time. Oh, so conclusion, not a frequentist. I'm a person who tries to understand the best tools for the problem. Bayesian analysis has many, many powerful uses. Just phase 3 trials are not where it belongs. And I'll quit there, and I'm sorry I yacked for only gave. Are there any questions or comments or anything? That makes sense at all? I know it seemed like 1,000 different topics, and again, it was 1,000 different topics. Thank y'all.